This lesson is about glycolysis, the first phase of cellular respiration. It is an anaerobic process, meaning it does not require oxygen. Now, from the name, you can tell what it means, because glyco means sugar or glucose, and lysis means splitting, glucose splitting, and that's exactly what it does. As you see down here, you start with glucose, a 6-carbon molecule, which gets split into half to give you 2 3-carbon molecules. And it's the only stage of cellular respiration that takes place in the cytoplasm, mainly the cytosol, which is the liquidy part of the cytoplasm. Here at the left, you see glycolysis occurring. You have one glucose molecule, which gets converted to 2 pyruvate molecules, which are the 3-carbon molecules. So 2 3-carbon molecules which enter the mitochondria where there's oxygen and get oxidized into 2 molecules of acetyl-CoA, which then can enter the Krebs cycle. Now, there are 2 stages to glycolysis. First phase, which is the investment phase, steps 1 through 5, and second phase, the payoff phase, steps 6 to 10. Now, in the investment phase, what happens is that 2 ATPs are invested, but on the side of the payoff phase, what's produced is 4 ATPs, 2 NADH, and 2 pyruvate molecules. Now, even though 4 ATP are produced, only 2 are net because 2 were originally put in. It's like the stock market. If you invest $2 and then you end up with 4, you only net 2 because the first $2 were yours, but you originally invested. So you really made $2. And here the reaction on the bottom, chemical reaction for glycolysis, is right here. As you can see, glucose gets oxidized, but what do you think happens to 2-NADH? I mean 2-NAD plus. What happens to this molecule? Would you say it's reduced or oxidized? It's reduced because it was neutral. It was positive, and now it's a neutral molecule. 2-NADH are neutral because a hydride ion was added, which is a hydrogen atom with an extra electron, so it acts as an electron, and gaining of an electron is reduction. Okay, so to recap, 2 ATP molecules are used to split glucose, 4 ATP molecules are produced, but only net 2 ATP, 2 molecules of NADH are produced, 2 molecules of pyruvate are produced. As you can see, glucose splitting the glucose to 2 3-carbon sugars, and which eventually get turned into 2 pyruvate molecules. And here you see the steps. On the left is the investment phase, and to the right is the payoff phase. Now, in the investment phase, we start with a glucose molecule, and we have an enzyme called hexokinase. Hexo because this is a 6-carbon, hex for 6-carbon molecule. And then kinase is an enzyme that phosphorylates. It transfers a phosphate group to another molecule, from one molecule to another. So what it does is takes the phosphate here and transfers it onto here. And glucose is converted to glucose 6-phosphate, usually called G6P for its abbreviation. and that happens G6P here is changed into its isomer fructose 6-phosphate F6P. Now, as I told you in class, an isomer is a molecule with the same chemical formula but different structure. Same chemical formula but different structure. And as you can see, they both have 6 carbons, the same amount of carbons, oxygens, and hydrogens. So the only thing that's different is the structure. And that is done with the help of an isomerase, phosphoglucose isomerase. Isomerase, when you see ACE, you know it's an enzyme, and isomerase, it changes the structure, changes it to its isomer. And then what happens here with F6P is that it's actually phosphorylated. Here we have ATP again. So ATP is invested in step 1 and step 3, and with the help of a kinase, phosphofructokinase, because we have fructose that's getting phosphorylated. The name always gives you a hint. So the help, it's going to take the phosphate group from here and send it over and phosphorylate here. So you see what's changed. This got phosphorylated, and then it's converted into fructose 1,6 biphosphate, because it's carbon 1 here, which is phosphorylated, and carbon 6. So then what happens to fructose 1,6 biphosphate is that it's split into 2 3-carbon molecules. One that is called dihydroxyacetone phosphate, and then the second one is glyceraldehyde 3-phosphate, and that's what we want. This is the molecule we need. So we need 2 of them. So what happens is this one gets sent here and converted. The structure is converted by the help of an isomerase, changing it into G3P, glyceraldehyde 3-phosphate. So this is G3P, and so is this G3P. So we have 2 G3Ps that now can enter the payoff phase. And because we have 2 G3Ps, steps 6 through 10 occur twice. Okay, here you will see in step 7 where ATP is produced. In step 10, remember we have 4 ATPs, and because these steps happen twice in the payoff phase, we're really getting 2 of each here in each step, which totals 4. So here in step 6, G3P, what happens to it is get phosphorylated. Okay, you got a high energy molecule now with 2 phosphate groups. And then here, what it does is that it phosphorylates the ADP, converting it to ATP. That's where we get energy. Then what happens to 3-phosphoglycerate, because once 1,3-biphosphoglycerate had 1 phosphate group removed, it turned into 3-phosphoglycerate, and 3-phosphoglycerate here now is converted into 2-phosphoglycerate by a mutase, phosphoglycerate mutase, which changes the location of the phosphate group from the third carbon to the second carbon. And then 2-phosphoglycerate here, with the help of an enolase, it's called enol, the structure. So an enolase is the enzyme that converts it. What it does, it removes water and converts it to the structure called phosphoenol pyruvate. So we almost have pyruvate. Then this here is this phosphoenol pyruvate. Finally, it gets converted into pyruvate. And we get 2 ATPs here by the transfer of this phosphate group to ADP. That gives us ATP and pyruvate. And here you have the end results. 2 pyruvates, the NADH and the ATPs. Now, as we saw here in this reaction, some of the arrows are going in one direction and some are going in 2 directions. Like, for instance, here, it's going in 2 directions in step 2. But you see, step 1, it's going in 1 direction. What do you think that is? Remember I said in class that when a reaction is coupled with ATP, it will go in the forward direction. An energetically favorable reaction goes in the forward direction. So here you see there's ATP, it's going in the forward direction. And also step 3, there is also ATP, it's also going in the forward direction. Okay, because the ATP helps drive it to the forward, makes it go forward, and that is energetically favorable. So if you couple a reaction with ATP, it will make it go in the forward direction. Or if you add more reactant, increased reactant, will do the same. Or reduce product. Reduced product will also make it go in the forward reaction and be energetically favorable. So here, because ATP is already present, it is moving in the forward direction. Practice questions. Number 1, glycolysis is an anaerobic process used to catabolize glucose. What does it mean for this process to be anaerobic? You can pause for a second and think about it. A, no oxygen is required. In the glycolysis pathway, 1 blank is converted into 2 pyruvate molecules. 1 molecule of glucose. What is produced during glycolysis? B, for ATP, 2 NADH, 2 pyruvate. What is the net result of glycolysis? C, 2 ATP molecules, 2 NADH, and 4 pyruvate. Even though 4 is produced, because 2 is invested, so we really only net 2. 5. In which steps of glycolysis is ATP invested? Well, it's definitely in the investment phase, first of all. So you can cross these out. And it's 1 and 3, so it's C. In which steps of glycolysis is ATP produced? It's definitely in the payoff phase. Cross that one out. And 7 and 10, so let's see. How many times does the payoff phase occur from one glucose molecule in glycolysis? It's 2 times, because you get glucose splits into 2 molecules of pyruvate. Splits into 3 carbon sugars that enter the payoff phase to give you the end result of 2 pyruvate molecules. When a molecule of NAD gains a hydride atom in glycolysis, the molecule becomes reduced because it was positive and now neutral. Remember I said the hydride ion is a hydrogen atom with an extra electron, so it acts as an electron, and gain of electron is reduced. That's it for this video.